Hello. I am not visible, right?
Yeah.
PPT is visible, but I am not visible. Just a minute. Give me 1 minute. Some setting problem is there. Why it is not? Don't know what happened. Just a minute. Should come here. Why not coming here? Oh. Don't know why it is not coming. Without my face, you just listen to me. Okay. Just a minute. Why it is not coming? Plus. It is not. In this position, there should be pic. Don't know what is happening. What can I do? Hey Bhagwan. It should be visible. HP device. Yes, activate. Activate. Yes. Is it visible now? Lakshmi?
No, ma'am.
Yes, now I am there. See? Now it is visible.
Okay, yes, now.
Hmm. Now see. So today I'm going to discuss about the genomic and cDNA libraries. Okay. Now, as you know that from the eukaryotic gene, these are actually eukaryotic gene structure means here this is the DNA structure. Okay, in DNA you can see not only coding regions are present. Other than that, few other things are present like enhancer, silencer, promoter region, okay, and UTR region. So these are coming under regulatory sequence. We are talking about the DNA, eukaryotic DNA. Okay. Then one open reading frame is there. Open reading frame. Then open reading frame means then only intron and exon present in the open reading frame. Then again some regulatory sequences including UTR region and enhancer region, enhancer or silencer region. Okay. Now if you consider from the DNA generally pre-mRNA or hnRNA is produced. Okay, heterochromatin it's hnRNA. Heteronymic. hnRNA is produced. That means that is called the pre-RNA. Before the maturation of mRNA, it is produced pre-RNA. So in that case, what is present? In that case, mainly we are talking about the RNA part. That means intron, exon, and a few UTR region. Okay, UTR region here, UTR region here. Okay, so now just a minute, I should take a separate color. This color. Okay. So in pre-mRNA, actually intron present, exon present, plus few UTR region present. Okay. Blue color is for the UTR region. Now, when it is mature to mRNA, in mRNA only exon present and UTR region present. Right? Right? So if you consider about the mRNA, this is the mRNA, UTR region present, then 5 prime cap is added and poly A tail is added in the 3 prime position, 3 prime position, and this is the 5 prime position, right? This is the maturation process of the mRNA, and exon exons are integrated, where the introns are removed from the pre-mRNA by splicing process, right? And finally, protein is synthesized. From one mRNA, only one protein is synthesized, right? That is for the eukaryotic system. But if you consider about the prokaryotic system, in prokaryotic system, this is actually known as the polycistronic operon. In that case, regulatory sequence will be there, as well as open reading frame and UTR region. Here also same thing, regulatory sequence, ORA, UTR and open reading frame. But in the open reading frame, what is present? In case of prokaryotic system, there is no separation of intron exon. You can consider all are coming under the exon, no intron. Okay, so directly from the DNA, transcription is done to make mRNA. So there is no pre-mRNA during the prokaryotic transcription process, right? So in this case, you can see UTR region is present and mRNA is produced. Now from the mRNA, you can find from one mRNA you can produce several proteins. Therefore, it is known as the polycistronic mRNA. Okay, so these are two differences. These are the differences between prokaryotic and eukaryotic gene structure. Now, going to DNA extraction. So, why I am discussing these things? It will be easy to understand gene library and genomic library and cDNA library. Okay. Now going to DNA extraction and purification. Already I have discussed this thing. है ना? DNA, how to extract DNA and how to purify the DNA. Several methods I have discussed. Several steps already I have discussed. Uh, One popular method, this is conventional methods involving phenol chloroform extraction method, and another method is there that is involving absorption chromatography using silica gel. Okay, although nowadays kit is available, column is available, okay, you don't have to do anything. See, purification kits are available from multiple manufacturers. Okay. No, you don't have to understand this thing. Already I told you this is the phenol extraction process. If you add phenol and chloroform, what will happen? In the aqueous layer, you can collect DNA and RNA, but in the interface, there will be coagulation of protein and phenol will be lower at the lower part. In this way, you can separate DNA RNA from the protein. And finally, you can isolate the DNA by adding ethanol. Another way is the this is the absorption silica method. If you put the DNA in the silica gel, DNA will bind with the silica gel. You can isolate that. Okay, nothing else. Now we generally check purity of the DNA by checking this value 260 divided A260 divided by A280. है ना? It should be higher than 1.8. Okay, and another 230 value is there. Okay, that is for protein backbone and salt phenol. They can give you these few peaks at 230 nanometer. Okay, because phenol extraction method is used now. If you are using phenol extraction method, phenol may give you peak. Acha, this is actually known as the denaturation curve. Okay, TM means temperature of denaturation. In this way, you will get curve. In x-axis, it is temperature. In y-axis, it is 260. Why? When the temperature of double-stranded DNA solution is increased in some range of temperature, the A260 increases by 30%. So generally, see from this section, it is increased. Okay, then after that, it will be stable. Now, this is called the hydrochromic effect, which occurs when the bases in DNA unstack on denature of double-stranded to single-stranded DNA as the intra-strand hydrogen bonds break. So, denature means double-stranded DNA is converted to single-stranded DNA. Okay, because hydrogen bonds are broken. So, in this case, what is happening due to the denaturation? A260 value is increased. Okay, A260 is mainly for the protein you can consider. No, that is for the DNA. It is increased. Now see, now The midpoint of the cooperative change in the absorbance signifies that the population of DNA molecule is 50% denatured. So from this graph, we generally measure the 50% denaturation temperature. So this is called the 50% denaturation temperature. How you have to measure A260 value versus temperature value. Okay. Okay. So nothing else. Now going to the main thing, that is the gene library. Gene library from the name you can understand ki a lot of things are collected there. Now gene library means construction of cDNA library and genomic library. What is this? Oh, this is the content. What are the things present here? You don't have to understand this thing. Now see, you have to start from this slide. So two libraries possible. From the name of the topic, you can understand a genomic library and cDNA library. That means you can construct two types of libraries, cDNA library and genomic library. cDNA, that means you know na complementary DNA. Complementary DNA is produced from where? From the mRNA. Okay, so genes in expression, that is mRNA, you have to make it to cDNA by reverse transcriptase enzyme. The starting point is the single stranded DNA, single stranded cDNA, then double stranded cDNA formation. And now you just make plasmid. You just construct the recombinant with this cDNA. Now, if you consider the genomic library, genome means all total DNA, complete DNA set, chromosomal DNA. Okay, now you just digest it. Okay, do the digestion by the restriction digestion, endonuclease enzymes. Now, all the gene fragments you make separate recombinant. Okay, so that is, so these are known as the library, cDNA library and genomic library. Because of genomic library, you want to make the genomic library, you have to digest a bigger size genome into small fragments and make separate recombinant by paste. You have to paste it with a plasmid. So cloning vector, you have to clone it here. Okay. So multiple copies of recombinant sets will be there in the library. Now size of the library. Ensure enough clones means must contain a certain number of recombinants for there to be a high probability of it containing any particular sequence. That means you have to make a lot of numbers of recombinant things. Okay. Now how to make this? A formula. There is a formula. The formula to calculate the number of recombinants. How many number of recombinants are produced? This is the formula. Capital N is equal to ln 1 minus P divided by ln log 1 minus f. That means number of recombinants, how much amount of number of recombinants are produced, that is equal to ln into 1 minus p. P is the desired probability, whereas f is the fraction of genome in one insert. Fraction of genome in one insert. That means if you consider that this is the total genome structure, na? This is the total length. Okay, if you consider this is 100 kb, I'm just example I'm giving kb. Now if you have constructed library, or you have constructed recombinant here, only this part. Okay, so this is only you just consider the 10 kb. Okay, 10 kb fragment. So f means the fraction of genome in one insert. So you will insert this part na, in order to make recombinant one. So what will be the fraction? It will be 10 by 100. Okay. So it will be 10 by 100. In this way, you just remember the formula. N is equal to ln 1 minus p divided by ln 1 minus f. Okay, desired probability f is the fraction of genome in one insert. Now, genomic library means set of clone fragments that collectively represent the genes of a particular organism. Okay, so genome I told you several times, genome means whole set of gene. Okay, whereas proteome means whole set of protein. I told you na. So genomics means we have to study whole set of genes. Okay, so you have to make clone. Uh, you have to make different recombinants from one particular genome, from one particular organism. Okay, so a gene library is a set of cloned fragments. Clone fragments. So first of all, you have to make fragments, na. Now make the fragments by adding restriction enzymes and separately. You just clone all the fragments. Okay. Now, particular genes can be isolated from the DNA libraries. So, whatever you want to, uh, suppose ki this part of the genome you need. Okay. So, you can collect that recombinant. You can collect that recombinant and use it. Collect that recombinant and use it. So particular genes can be isolated from DNA libraries. Both genomic and cDNA libraries can be screened by hybridization process. So during the making of library, there will be mixture of fragments, okay, mixture of recombinant. So in order to identify your gene of interest or gene of interest, you have to use this screening method, screen by hybridization technique. How it is happening, let's see, is there anything informed here or not? Let's see. Now, what is cDNA? So here it is the genomic library. Genomic library, whole genome, you are just making the fragments, making recombinants, and then you just your interest of gene you can collect from the library by hybridization technique. Now going to cDNA. cDNA complementary DNA libraries are generated by the reverse transcription. Already I told you same thing. Now, cDNA is representative of the mRNA population. Why representative? It is just complementary to mRNA. Na? And in this case, in the cDNA, there will be intron or not. Intron sequence will be present in cDNA or not? Tell me. If you consider about the eukaryotic system, if cDNA is the complementary of mRNA, then whether there will be any
No,
intron.
ma'am. It will not be present.
Only exon and UTR, right? So, and therefore reflects mRNA levels and the diversity of splice isoforms in particular tissues. Okay, so now mRNA cloning not possible. Now I told you mRNA cloning means you have to convert it to cDNA, then you have to make it double stranded DNA, and then only you can insert into the cloning vector. So in order to do that, you have to make the library of cDNA libraries. Now the PCR can be used as an alternative to cDNA cloning. Okay. Now full length cDNA cloning is facilitated by the rapid amplification of cDNA ends. So there is a method, RACE is the method. I think it is PCR based method. From that you can make cDNA clone. So full length cDNA cloning is facilitated by the rapid amplification of cDNA ends. That is race. I have to check what is race. I forgot it. अच्छा. Same thing क्यों बारे बारे क्यों लिख रहे हो भैया? So this is written that intron is not present, lack introns. Okay. Now introns are rare in bacteria but occur in most genes of higher eukaryotes. Achha. Now introns interrupt the collinear relationship of the gene and its encoded polypeptide. Introns may occur in the. What is this? It is not required. Now, size of the cell DNA is significantly smaller than that of the corresponding genomic. Okay. Now this one. So obviously, size of the cDNA clone is significantly smaller than that of the corresponding genomic clone. Obviously, na, I have shown in the eukaryotic system here that if you make genomic library, so you have to construct it from the DNA, whole genome. So that size will be higher than this mRNA, है ना? Because you are removing all the, at least you are removing the intron part. So initially genome, if you consider this is the structure of genome, mRNA, if you full length you just measure, it will be just like smaller than full genome. So obviously, whenever you are making cDNA library, it will be smaller, है ना? Smaller fragments will be there, smaller sized library will be there, whereas as compared to the genomic library. So that is given in this slide. A size size of the cDNA clone is significantly small spelling देखो, smaller than that of the corresponding genomic clone, है ना?
Yes, ma'am.
Now see, example is given. So human dystrophin gene contains 79 introns. Okay, so if you remove this part, so corresponds over 99% of the sequence, representing over 99% of the sequence. The gene is nearly 2.5 mega base pair in length, and yet the corresponding cDNA is only 11 base pair. So total length of the gene, it is 2.5. It is whereas cDNA is only 11 base pair, okay, smaller than the gene. Now, cDNA clones can be expressed in E. coli since removal of eukaryotic introns by splicing does not occur in bacteria. Okay. So, this can be done to locate intron exon boundaries in eukaryotes. Okay. Can be expressed in E. coli since removal of eukaryotic introns by splicing does not occur in the in bacteria, because only in exons are present in the cDNA, so you can express it. There is no requirement of the post transcriptional modification inside the host cell. Okay. Now, construction of gene libraries कहाँ से करते हैं? A DNA library of or gene library is a collection of DNA clones gathered together as a source of DNA for sequencing, gene discovery, or gene function studies. So these are the purpose why we need DNA library. Now, depending upon the DNA source, library can be grouped as genomic library or cDNA library. Okay, depending on the DNA source, library can be grouped as genomic library or cDNA library. Now, genomic library are produced when the complete genome of a particular organism is clipped into thousands of fragments, and all the fragments are cloned by insertion into a cloning vector. Okay, so that is the there. Why several times they are telling. Now, first step is you have to make digestion. You have to do the digestion by restriction endonuclease. Okay. Now, next is the fragments that are too large or too small for cloning are removed by centrifugation or electrophoresis, and cloning vectors such as BAC or YAC plasmid, those are clipped with the same restriction endonuclease and ligation to the genomic DNA fragments. So this is about the cloning, how cloning is done. Actually, same restriction enzymes are utilized for the cloning vector as well as for the genome interest. So here same thing is written. In this case, if you are using eukaryotic system BAC, or you can use YAC plasmid, you just insert all the fragments, so you can get multiple recombinants for multiple fragments. Okay, and the ligated DNA mixture is then used to transform bacteria or yeast cells to produce a library of cell types, each type covering a different recombinant DNA molecule. है ना? Whenever you are transferring to the host cell, that means a BAC or UAC, what will happen? Cell expression will be different. Expression will be different for a different recombinant part, recombinant plasmid. Okay, so ideally all the DNA in the genome under study will be represented in the library. So ideally all the DNA in the genome under study will be represented in the library. So what is happening? See, again I'm reading this line. The ligated DNA mixture is then used to transform bacterial or yeast cells to produce a library of cell types. Each type hovering are different recombinant DNA molecule, right? Because in the first slide I have given now, you are making, this is the one genome. You are making different fragments, right? If you consider this is one fragment, now if you, this is another fragment, if you consider another color, just giving you another color, this one. Okay, suppose three, more than 3,000, above 1,000 you can make. Now if you just consider that this genome is converted to these three fragments by restriction endonuclease, you have to construct the recombinant one. How? You have to insert these fragment into, where you have to insert into plasmid, right? So you just add the plasmid, you just construct recombinant, separate recombinant you can make, separate recombinant you just make. That means you are inserting these fragments into separate plasmids. Okay, now another, you, from these three fragments you found three recombinant plasmids. Now you have to insert these three. Okay, you have to insert these three. Where you have to insert? In separate. What will happen in separate bacteria or BAC or YAC yeast cell? Those are the host cells. So you just insert one by one. You just insert here, one molecule will be inserted here, and another recombinant will be inserted here. So in the mixture, you can find various host cells with various recombinant plasmid, right? Finally, you are getting various recombinant plasmid they are present inside the host cell. So various host cell expression will be there here. So that is given here. The ligated DNA mixture is then used to transform bacterial or yeast cells. That means BAC or YAC generally used to produce a library of cell types, each type hovering at different recombinant DNA molecules. So if you consider this will give you one recombinant, this will give you another recombinant, and this will give you another recombinant. So all together you are making the library. Okay. Now cDNA, what are the steps involved here? अरे भाई कहाँ है? Same thing है यहाँ पे भी दे दिया. Whatever it is mentioned in this figure ना, first figure. In this figure it is given to genes in expression mRNA. It is converted to cDNA, that is the single stranded DNA, and by the reverse transcriptase. Now single stranded DNA is converted to double stranded DNA by the replication. Okay, and finally you have to construct the recombinant by joining it with a plasmid. Okay, and now transfer it to any host cell, nothing else. Now see, the synthesis of double-stranded cDNA suitable for insertion into a cloning vector involves three major steps. First strand DNA synthesis on the mRNA template by reverse transcriptase, then removal of RNA template, then second strand DNA synthesis using the first DNA strand as a template by DNA polymerase. And all DNA polymerases, whether they use RNA or DNA as the template, require a primer to initiate the strand synthesis. So during double strand synthesis, primer is required. It is there. And with the help of DNA polymerase, what will happen? There will be synthesis of double stranded DNA. Now, Acha. So in this case, what is happened? Primer. What type of primer is utilized? If you are taking mRNA from where? If you are taking mRNA from the eukaryotic system, so what will happen? Poly A tail will be there, है ना? So if poly A tail is there at the 3' end, so you can add primer as the poly T or oligo T, है ना? So easily you can construct this primer. So now there will be replication. So in this case, you just add oligo DT primer in order to make double-stranded DNA. So this involves the use of an oligo DT primer annealing at the polyadenylated tail of the mRNA to prime first strand cDNA synthesis. Okay. Now after the synthesis of second DNA strand, this loop must be clipped with a single strand specific nuclease. And so finally this type, so I think I have given this type of figure already. I have discussed this thing. So finally last part will be degraded by the S1 nuclease. So let's see from this figure also it will be completely clear. This method already I have discussed. See, this is the first step, mRNA, and now oligo T is attached so that reverse transcriptase can make reverse transcription can make what? Single stranded DNA. Okay, first strand synthesis is happening in this way. This is the single stranded DNA. Okay, because here poly T is present, na? So this is the single strand. Now DNA polymerase, what will happen? DNA polymerase synthesis will help to make the double strand. So double strand means in this case na 5' to 3' position. So from here it should be added. Na? It should be 5' to 3'. In this way synthesis will done in the replication process. So it will be, so what will happen now? This part is actually joined together. Okay. So in order to degrade that, S1 nuclease is involved. So that part will be degraded by the S1 nuclease so that you can separately get this type of double stranded DNA. Okay.
Ma'am, which part is degraded by S1 nucleus?
See, in this case, now just I'm showing it, drawing separately so that you can understand. Now, if you consider this is the mRNA, initially it was mRNA. Okay. Now giving you new thing, that is you are making one cDNA. cDNA, that is the single stranded one. Now the single stranded DNA, so there will be this type of one loop will be there. So this is from this is at the 5' one. Okay, at the 5' one, and this loop actually it will continue to make the double strand by the polymerase enzyme. Okay, so you will get this type of formation, loop type of formation. Now S nuclease will degrade this part, because in order to get the double strand you have to be this type of, na, just parallel line will be there. There should not be joining of that line. This bond should not be there. है ना? Joining is not there. This type of joining should not be there. So it is degraded. This part is degraded. See this part. So after the double strand formation, S nuclease is degrading, joining between the double strand. Not hydrogen bonding. Diester bond is degraded. See, after the synthesis of second DNA, this loop must be clipped. So what loop? See, why loop is formed? Is it given? Took advantage of the fact that the first cDNA strand has the tendency to transiently fold back on itself, forming a hairpin loop. Okay, cDNA has the tendency to make the hairpin loop, resulting in self-priming of the second strand. Okay, self. Acha, 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 acha. See, what is happening? Initially, in order to make single strand cDNA, you need oligo T primer. Okay, So first step you need primer, but in the second step when you are doing the replication in order to make the double stranded DNA, at that time you don't need any primer. cDNA itself is making a loop in order to synthesize second strand. Okay, so what is happening here? If you again, I'm trying to clear it. So if you consider this is the mRNA, okay, so poly A tail is present here, right? Poly A tail is present here. Now against poly A tail, in the first step, you have when reverse transcription is happening, at that time you have to add oligo T. Okay, so if you are adding oligo T, so then what will happen? This type of synthesis will done. Okay, complementary to mRNA, so synthesis is single stranded synthesis. So this is actually the complementary DNA initially. This is cDNA. Okay, so this is happening due to the reverse transcriptase enzyme. Now cDNA, it should be converted to double stranded. How it is happening? See, actually cDNA, so in the next step this RNA is degraded by the RNAs. Now cDNA is free, right? Now cDNA is free. In this way, if you write, it will create one hairpin structure in this way. Clear? If it is making the hairpin structure, it is actually creating the primer. So this part is, if you consider this is the 3' part, okay? So, and this is 3', and this is 5', right? This is 5'. Why? Always synthesis is happening from the 5'. So this here T part was the 5'. T part was 5', and here it is the 3' end, and 3' end is actually folded again. It is actually folded, right? If folding is happening, so small part is folded. So this is actually making the primer. It is acting as the primer. So what will happen? There will be no requirement of addition of any other primer. Okay, so there will be bonding in between these. So this part is actually acting as the primer, folding part. If it is acting as the primer, very easily DNA polymerase can synthesize what? From 5' to 3' region, it can synthesize what? DNA second strand here. So now you have double stranded DNA. Double stranded DNA, but with the hair loop structure, this hairpin structure. Now coming to the S1 nuclease. S1 nuclease will degrade this loop structure. Okay. Whenever the loop structure is degraded, now you have this double stranded DNA cell. नीचे part blue में रखते हो, ऊपर वाला part green में रखती हूँ. So now you have this double stranded proper DNA जो होता है ना strand fragment जैसे होता है, without folding anything, you can get this type of fragment. Clear? Now is it clear?
Yes, ma'am.
है ना? Folding, why folding? It has a tendency, cDNA has a tendency to make the folding. Why folding? Because hydrogen bond is possible, maybe in at this part complementary sequence is there at this position and below position, some complementary sequence will be there, therefore they are making this type of hairpin structure. Okay, so it is written cell priming, so there is no requirement of another primer. Primer is always required na, for the DNA polymerase. In the first step in reverse transcriptase, okay, that is actually RNA dependent polymerase. So reverse transcriptase, in that case, primer is required. But in this case, DNA polymerase, DNA dependent DNA polymerase, in that case, cells priming is happening. Only in this case, not always. DNA polymerase, it also, by using the primer, it will increase the length of the DNA or it will replicate the DNA. Okay. Okay. Chalo. So I hope this is clear. Now going to next, anything else is there. Now there is another improved method. Improved method for cDNA cloning. How it is happening? A serious disadvantage of the hairpin method is that cleavage with S1 nuclease results in the loss of certain amount of sequence. Obviously, whenever this type of loop is formed, so this, if you cut this part, so there will be removal of some sequence, है ना? Loss of the sequence, loss of certain amount
Yeah.
of sequence at the 5' end of the clone. Now, this strategy has therefore been superseded by other methods in which second strand is primed in a separate reaction. So in this case, second strand is primed. That means some other primer is added. Okay. So first strand is tailed with oligo DC. So in that case, whenever this cDNA, single stranded cDNA is produced, single stranded cDNA is produced, what is done? First strand is tailed with oligo DC, allowing the second strand to be initiated using an oligo DG primer. So in this case, you have to add some oligo DC. DC, you have to add DC. Okay, DC. So opposite to DC, complementary structure is the DG. So you just add, you have to add oligo DG. So oligo DG will bind with this C. So there will be primer G. I'm just writing G. Okay. So this part is the primer one. You have to add separately. So before adding the primer, you have to add oligo DC so that it can bind at the 3' position. Okay. 3' position of single stranded DNA. Okay. Now see here what is happening. Up to this, it is uh, same to same. So mRNA now single stranded cDNA.
Ma'am, the video is free. Join again.
Oh,
Hello.
okay, okay, join.
Leave and join again.
हाँ okay. So in this case, what is happening? Poly C is added at the 3' position. So against that, you can add the primer oligo DG. So oligo DG will give here, it will bind and it will create the double strand. Okay, so bonding, there will be primer bonding here, and now synthesis is happening from the 5' to 3' region. So now you can make the cDNA, a double stranded DNA. So there is no loop formation. We have restricted the loop formation by adding CC region. Okay, DCC region, DNA as DNA, so it is DCC. Okay, now insert into vector. Now finally this double stranded DNA you just insert into the vector. Okay. Achha. Now, okay. Now methods for directional cDNA cloning. Is there any? Just a minute. I have to make it slightly different way so that you can observe it. An early stage strategy in which the formation of a loop is exploited to place a specific linker in this example is say to at the open end of the duplex cDNA. Following this ligation, the loop is clipped and trimmed with S1 nuclease and EcoR1 linkers are added to both ends. Cleavage with EcoR1 SA1 generates a restriction fragment that can be unidirectionally
Ma'am, the
inserted
video
into a
is not
vector.
freeze. Ma'am, we are in which slide?
This is the slide number 24.
Okay, now okay. It was interface with 23,
Okay.
facing a lot of network issues, or I don't know, it's the app or network.
I think it's app. This class plus is not. Uh, Whenever you are joining from the G Meet, Google Meet,
Yeah,
there was no
was
such
no problem.
problem. No?
Mm, mm.
Uh, Still, it is not visible.
It's like visible for some time, then it gets freeze. Now again it's freeze.
You are using Wi-Fi or you are from data? You are in
I
mobile
tried
data.
Wi-Fi also and mobile also. I'm trying both of them.
Okay. You have Wi-Fi of Jio or something else? Is it from Jio or Airtel?
I Wi-Fi is this they provide.
Don't know.
I think it's zero.
So Jio connection is
Wi-Fi.
nowadays good. Okay. Whenever it is clear, you tell me. I will resume it. Hello. Hello.
Mm,
Huh. Is it visible? No.
no, no,
I am showing 23. ये भाई क्या करें क्या करें that student is not that your friend is not joining today. Shraddha is not joining today. Don't know. Yesterday she told me ki she will join, but she couldn't join. Okay, is it visible now? No.
it got freezed again. Ma'am, can't we do in Zoom meeting?
अरे यार, they strictly instructed me to do it through this. Otherwise, I used to take from the Proctor app. Proctor app is through the Zoom only. It was better than this class, I think. Actually, this is actually live streaming is happening. So together with I think OBS, another software is added with it. Therefore,
Okay, now
creating
it's
problem.
visible.
It's visible. Okay, now see.
Mm,
In this case, you are making the duplex cDNA or double stranded cDNA, right? Now, when this is actually blunt end, right? Most of the cases we insert any DNA with the cohesive end. That is better, na? Cohesive end means some hanging region will be there at the 3' position, है ना? So
yes, ma'am.
two sides of the position. So in order to make that cohesive end, you can add a linker. Okay, so that is done in this slide. It is given this way key how you can make the cohesive end. Okay, so in this case, what is happening? An early strategy in which the formation of a loop is exploited to place a specific linker at the open end of the duplex cDNA. Now, following this ligation, the loop is clipped and trimmed with S1 nuclease and EcoR1 linkers are added. From the picture, it will be clear. I am just reading it. linkers are added to both ends. Now cleavage with eco R1 and SAI1 generates a restriction fragment that can be unidirectionally inserted into a vector clipped with the same enzymes. Now we are going to observe what are the strategies used in this first process. So from this picture it will be clear. Okay. So if you consider this is the mRNA, from mRNA you have constructed, you have added poly T primer so that you can make cDNA. Okay, due to the presence of the reverse transcriptase enzyme, it is making the cDNA. Now cDNA is making that loop structure here, hairpin loop structure. Now, whenever you are adding the DNA polymerase, you are not adding S1 nuclease. Okay. In this case, you are not adding S1 nuclease. You are adding polymerase and you are adding. So, what if you are adding the polymerase, what will happen? This type of double stranded structure will be there. Hairpin structure, full hairpin structure. Now, you just add linkers. S linker means in this linker there is restriction site for Cy1 restriction endonuclease. Okay, so you just add it, that sequence. Okay, now you have to add the, after that you add nuclease S. Now you just add the nuclease S1. So what will happen? This hairpin structure will be degraded. Okay, now you have right side of the position, you have that Psi1 restriction endonuclease site. Again you have to add eco R1 linkers. That means some sequence you have to add where in that sequence eco R1 restriction site is present. So at the opposite side you have to add the R restriction site, eco R1 linker. Okay, in this case both of the sides it is added, left side as well as right side. Okay, now whenever you use eco R1 plus psi1 restriction endonuclease, what will happen? There will be cut here, one cut here, here, these are removed and you can get this type of cut. Okay, here, so you can get this type of cohesive ends. Now you can get the cohesive end. Now you can insert it into the vector. And vector, where vector also should be degraded with the psi1 and eco R1 restriction endonuclease. Then only this part will be annealed together. है. So that is the case happening.
Mm.
Okay, now the second process. Second process, what is given? A similar strategy, but second strand cDNA synthesis is random primed. And the oligo-DT primer carries an extension forming a Psi1 site. During second strand synthesis, this forms a double-stranded Psi1 linker, and the addition of further eco-R1 linkers to both ends allows the cDNA to be unidirectionally cloned. So let's see from the picture whether we can understand or not. Let's see. First of all, add oligo-DT primer with Psi1 side. So you have to add DT, you have to add oligo DT. Along with that, you have to add one Psi restriction site. Okay. So what will happen? Reverse transcriptase will make transcription. cDNA. So this is actually cDNA. Now DNA polymerase. Okay, you have to add DNA polymerase. Then opposite to this cDNA, there will be synthesis of but why here no hairpin structure is happening? Can you understand? In this case, hairpin structure is not formed. It is not formed. I don't know why. So DNA polymerase, if you add, it is the
I think
just
because
huh?
we have I think SA1 we have already added in the first one. I think because of that.
No. See, we are making this. This is So this part is open, na? This here also this part is open. This part is open. It should make the hairpin, but in this case it is not happening. Don't know why. Let's see. DNA polymerase plus. So what will happen? Synthesis is happening. Second strand synthesis are happening. Oh, simultaneously in order to restrict the hairpin, na? The DNA polymerase along with the eco linker is also provided, so that instead of hairpin, R structure is added. This R means eco R1 linker is present. In this linker, eco R1 restriction site is present actually. Some sequence is added here at this position. Okay, so left side as well as right side. So everywhere, whenever eco R1 site is added, both of the sides it is added. Okay, now Psi1 and eco R1 digestion is done. So you can get this type of, same type of ending. कोहेसिव एंड ओके सो व्हाट आर द डिफरेंस डिफरेंस क्या है इसमें दोनों में क्या डिफरेंस है क्या डिफरेंस है देखते हैं इको आर वन बाद में डाला गया है इसमें है ना इसमें इको आर वन को बाद में डाला एस वन से पहले डाइजेस्ट किया बाद में डाला और यहाँ पे देखो इमीडिएटली आफ्टर द पॉलीमरेज इको आर वन को डाल दिया तो क्या है हेयरपिन स्ट्रक्चर इज नॉट फॉर्म है ना we are restricting the hairpin structure formation by adding the linker ठीक है okay so in this way now screening of the gene libraries why this type of structure is coming अच्छा screening gene library or genomic and cDNA by nucleic acid hybridization you can identify nucleic acid hybridization. Most commonly used method of library screening because it is rapid. It can be applied to very large numbers of clones, and in case of cDNA libraries, can be used to identify clones that are not full length and therefore cannot be expressed. Okay, so nucleic acid hybridization means I told you about the hybrid. So if this is the length of the DNA you want to identify, you have to add probe with labeled one. है ना short strength DNA with labeled compound अच्छा now screening by PCR you can do also you can screen the recombinant one by PCR also it is possible if there is sufficient information about the sequence to make suitable primers. Oh, PCR screening is only possible if you know to some extent about the sequence. Then only you can construct the primer. A lot of companies are there. They can make primer. Okay, they can construct you. If you give the sequence, they will make you primers. Okay, give you primer. So in order to make the primer, you have to know some sequence. Then only possible. Now screening expression library. In that case, if a DNA library is established using expression vector, if a DNA library is established using expression vectors, each individual clone can be expressed to yield a polypeptide. Obviously, if you want to observe the expression, so in that case, screening expression libraries. So if a DNA library is established using expression vector, I told you cloning vector, expression vector difference is there. Expression means something peptides or proteins should be produced. Okay, in that case, you have to use the expression vector in order to make the library. Okay, now nucleic acid hybridization technique was developed during. I think I have to take separate class for this chapter. It's not good. This PPT is not good actually. As a nucleic acid hybridization was first developed by who? Grunstein and Hognes, and it is modified by Hanahan and Messelson. Okay. Now in this case, what is happening? Colonies can be screened by the replica plating. Okay. I told you about the replica plating process. I know. The colonies to be screened are first replica plated onto a nitrocellulose filter dish that has been placed on the surface of an agar plate. And in that case, you should have one reference set in order to compare two plates. Therefore, only it is known as the replica plate. Now, colonies on the master plate is retained. So this is only given about the replica plating. Now the filter bearing the colonies is removed and treated with alkali so that the bacterial colonies are lysed and the DNA they contain is denatured. Now you just take all your required strain from the plate and you just digest it, and or cell membrane digestion is done, alkali treatment is done. Now what you have to do? You have to isolate the DNA. Okay. Now filter is then treated with proteinase K to remove the protein so that you can just get the DNA only. To remove the protein and leave denatured DNA bound to the nitrocellulose, for which it has a high affinity in the form of a DNA print of the colonies. Okay. So that means whenever on the nitrocellulose membrane, due to the replica plating, what will happen? If you consider this is the plate. Okay. Now your interest, recombinant strain colonies are there. Okay. On the plate, you have to digest it. You have to remove the protein, cell debris, everything. So in this case, what will happen? Only denatured DNA will be there attached with the membrane. Denatured DNA will be there attached with the membrane. Those DNA is actually representing the cell. Okay. Now, DNA is fixed firmly by baking the filter at 80 degree. The defining labeled RNA is hybridized to this DNA. Now you just do the hybridization. Hybridization is like which blotting? If you want to identify the DNA,
Southern blood.
southern blotting. So hybridization means you have to do the southern blotting only. Okay. And at that time I told you na ki single stranded DNA can only bind with the probe. So that is happening here in this process. So now the colony whose DNA print gives a positive autoradiographic result can then be picked from the reference plate. Okay, so from a library you can in this way select your protein of interest. Your DNA of it. First of all, you have to select the recombinant cell. Okay, and then from that you just identify the gene by adding the probe. Okay, that is called the hybridization technique, exactly like southern blotting, whatever. Acha. So this is the method how it is given, replica plating, how it is happening. Okay. From where I should start transferring? Is it's not visible, na? Time is over actually. Should I continue it in next day? Huh?
Yes, ma'am.
है ना? Slightly this PPT is not good. I think I have not observed properly. I thought it's good, shortly explained, but it's not good. I think lot of faulty results are there. I'll try to give you a separate PPT. Okay, so that it will be easy to understand. So main thing, library means you are making a lot of recombinant cells, right? What you understood from the topic? That is the main thing. If you are making genomic library, your starting point will be from the genome. You have to digest it, you have to make the fragments, and then construct different recombinant vector, and you just transfer it to host cell, right? So host cell will be recombinant cell, different recombinant cell. So those, if you collect every all the recombinants, that is the library. Okay. Now, whereas if you consider the cDNA library, that means you have to make it double-stranded DNA, and then you have to insert it into different vectors. Nothing else. Different mRNA, different cDNA, and different double-stranded DNA. Now they can be um, uh, inserted into the vector, so you can get a lot of cDNA library, cDNA recombinant. Yeah. है ना, that is the main thing, whatever I have discussed, nothing else. That is the main thing. And how you can identify, if you, a lot of recombinant cells are there, you want to identify only your protein of interest or your DNA of interest, not protein, DNA of interest. You have to choose, by most of the cases hybridization technique is the main process, and other processes also I will discuss next class. Okay. ठीक है ना?
Okay.
Okay. Acha, whatever I shared now, a lot of videos I have shared, that is from flow cytometer previous year question. You must check it. If possible, you write down in your copy. If possible. Otherwise, you can just observe it, what they are trying to say. Okay. Most of the cases, you will see this type of, from the graph, you will get questions. Okay. Have you seen?
Okay.
Today, few videos I have shared. You try to understand the topic. Okay. Only question answer, not full topic. You don't have to focus on full topic. You can watch
Yeah.
only question answer session, है ना? चलो, today we can leave.
Okay, ma'am. Thank you, ma'am.